important 1999 paper, Claes and Hegel, in Journal of Biomechanics. They had first come up with the mechanorevolutionary principle of, it is based on strain stimulus. The strain stimulus is nothing but the octahedral shear strain, okay, in combination with the hydrostatic pressure of that particular tissue. So octahedral shear strain and hydrostatic pressure, the values, threshold values, together contribute towards the choice of what type of tissue is formed. Okay? Newly formed tissue there. Now, we had given a lot of thought about how to execute this computational study, which would be meaningful and which would be contributing towards the world literature. Okay. The main problem was mapping from a macroscopic model to a microscale model. The displacements, the boundary conditions, because boundary conditions play a very, very vital role in the whole tissue differentiation process. Okay. Now, from the full-scale model, I hope you remember the full-scale model of the pelvic bone with the implant. We had come down and made a sub-model, and we transferred the displacements at the cut boundaries. Okay. The main purpose was to focus our attention in the vicinity of the region of interest, because our interest is focused in and around the area where the implant is sitting. Okay, because all these failure, everything is happening. The big change is happening near the acetabular cavity. But now we have to really look more deep inside into the periphery, that is the implant bone interface boundary. Okay, so we require to actually map the displacement fields from this submodel to a microscale model. Now, obviously, I have shown one small element here at a location. So if I vary the location at different places, obviously the implant bone displacement would vary. And what would also vary is the bone material properties, because bone material property is highly heterogeneous and it will vary from location to location even within the acetabular cavity. So in order to take both the variations into account, first the implant bone displacement field and the second the bone material properties into account, we actually took into consideration about 13 models, 13, okay? So we'll go into that a little later, but here let us just summarize the features of the microscale model. So element edge length was about 40 microns. This is the total number of elements that used, 1,24,000. Around total nodes 1,71,000. Now, for this microscale model, the applied boundary conditions mapped from the acetabular side, the top surface, mapped from acetabulum, the top surface is the bone surface. So mapped from the acetabulum, that is where the bone is. The bottom surface is the implant, pure implant, so that is mapped from the implant. And the lateral surface has periodic boundary conditions, the same boundary conditions of the two lateral surfaces. Okay. So as I was telling you, in order to take into account the variation of implant bone displacement fields as well as the variation in bone material properties at the interface, we defined 13 regions of of interest. And we had labeled, color labeled it, red, black, blue, and purple, based on the amount of bone, percentage of bone ingrowth in these 13 regions. Okay, so if the bone ingrowth was 12 to 25%, we mark it as red. So in all these regions, red blocks, it was between 12 to 25%. In the black, 45 to 55%. In purple, 60 to 70%. And in blue, we found it only in 2 regions. It is 75 to 90% bone. Okay. So please carefully try to read this slide, that how we had taken into consideration the variation of bone material property and implant bone displacement fields at the interface. So how many regions are there for the red block? There is 5 regions straight away, and it is around 12 to 25% bone ingrowth. Okay. The black region is 4 regions, 1, 2, 3, and 9. It is almost double. Okay, 45 to 55, more than double. Not almost double. It's clearly more than double. Okay. For the purple one, only 2 regions, 60 to 70, and the blue one, that is 75 to 90. You see the locations are within the SW, where you have very high bone ingrowth. Now we take each one from this color band. Okay. We pick up 5, 7, 8, and 9. So 5, 7, 8, and 9, one of each color band. And we try to have a insight into the distribution of the newly formed tissue layer. Okay? So you can see very clearly that the granulation tissue layer has transformed into, the red is immature bone, the green is the cartilage, and blue. The blue you can see here. In region 7, as I'm pointing with my pointer, there are some blues here. Clearly you can see that. Okay. There may be slight blues here, but not much in region 8. Okay. More in region 7. 5, 8, 9 has, even 7 has a visible amount of bone ingrowth. Okay. So the location of 5, 7, 8, and 9 has been given in this figure. Now what we had observed for all the 13 regions, that the average Young's modulus of the newly formed tissue layer was observed to increase with time in all the tissue layers. That means tissue is being differentiated from granulation, very soft granulation tissue, into more harder tissues. And overall, the average Young's modulus was increasing. So if you take 5, 7, 8, 9 as representative regions, you can see how it has grown over time, iterations, and then stabilize. And the strains in the newly formed tissue layer in these regions were observed to decrease. Because if the Young's modulus increases, obviously the strains will decrease. Okay? And it followed the similar pattern as was observed by suction. That is another important contribution published in Journal of Biomechanics about this tissue differentiation behavior, and we have compared similar trends obtained between their study and our study. Now comes the important comparison. This was more, very trendy comparison, very, very qualitative. Everything is qualitative at the the end of the day, because we cannot have a direct one-to-one correspondence of the same patient being operated and the same patient being followed up with quantitative values of bone differentiation or tissue differentiation. So we had to rely on some histological study, clinical study, which had predicted, or not predicted, we have predicted, they have observed from measurements and histological radiographic studies to come up with certain values, quantitative values. Now, if we look at our prediction, the overall finite element model predictions of bone ingrowth averaged over 13 regions for a single acetabular component for a single patient, it was about 45%. That is our prediction. Okay. The clinical studies had predicted in 2013, they had used 76 different acetabular components, and they were of titanium acetabular shell. Ours is cobalt, chromium, molybdenum, alloy implant material. Okay, they have come up with an average value of 76. The variation was from 20 to 83. Ours variation from 13 to 88. So these values are not too different. Okay. Long back in 1993, there were some data available from clinical studies. The porous coated acetabular implant for 9 different acetabular components, 33% bone ingrowth was predicted. Or not predicted, observed. Which matches with our, definitely, these values say something about our quality of prediction that we are predicting using a novel computational framework for such a complex set of events. Now, what was the important revelation here is we also compared the two models, the phenomenological diffusion-based model versus the cell phenotype model. And one slide I have summarized the outcome. Okay. As usual, we pick up 5, 7, 8, and 9. And you can see the white one is the phenomenological model, and the orange one is the cell phenotype model. Okay. The percentage of bone ingrowth in 5, 7, 8, and 9. Just see how close these two predictions are. But remember, this is the final outcome after equilibrium has been achieved in the tissue differentiation procedure. So, tissue differentiation around the acetabular prosthesis after attainment of equilibrium. So, what we can conclude that they were very, very similar. The final outcome. But if you look at the evolutionary process, there is a distinct difference in the two model predictions. Okay. As you can see, that there is a lateral shift in the two models. This is a major difference, but otherwise, the progression of the model predictions are almost similar. It is moving in the same manner. If you are looking from iterations, detailed iterative steps, you see that it is basically moving in the same manner with a lateral shift between the two, almost similar to a lag between the two processes. So obviously, you can conclude that the cell phenotype model has a delayed response to the whole procedure of tissue differentiation. And quite obviously, the delay is due to modeling of so many events. Each and every cell type is modeled in combination with different coefficients that contribute towards the modeling process, and that together actually causes the lag in between the phenomenological and cell-phenotype process. The outcome as well as the nature of progression is similar to the shortcut method and the elaborate cell-phenotype. So the major findings were the overall tissue differentiation predictions of phenomenological and cell-phenotype specific algorithms were found to be similar. Remember the overall predictions. Cell-phenotype specific algorithms are of prime importance where the cellular interaction with implant surface is required to be studied. So the cell-phenotype specific algorithm has its place in different kinds of investigation. So when there is cellular level interaction that needs to be identified or investigated, it finds its application there. But the phenomenological model, being computationally less expensive, is a suitable alternative to the cell-phenotype specific algorithm in order to gain an insight over the periprosthetic tissue differentiation process, which we were originally interested. We wanted to know the quantitative values of the spatial distribution of bone ingrowth around an acetabular ring, and we have achieved it. Questions? Now, if you read these two papers, I will just project it. I will not. Just a moment. Yeah. Can you all see this paper? Hello?
Yes, sir.
Bone ingrowth around post-coated acetabular implant. A three-dimensional finite element study using mechanorelatory principle. Okay? So published in 2015. Okay? So I will upload this paper, and there is another paper which is the comparison. Right? It's also Spindler publication, Medical and Biological Engineering in Computing, 2017. So these two papers are sufficient if you look into the equations, and of course all these things that I had discussed, everything will be, you will find it similar. Okay? Just you have to get a feel of what is happening. Okay? Here is the mechanorelatory tissue differentiation algorithm. The tissue formation and degradation, mass. We're discussing everything here. Finally, the results. You can see here the lag. Okay. and of course a host of the different kinds of things. But if you see here, you look at this paper, tissue differentiation during transition period in region 5 as predicted by phenomenological model and the cell phenotype. See the differences very clearly. Okay, this is an intermediate step. 5, iteration 5, iteration 15, 15. See the difference. 25, 25. At equilibrium, less differences. Okay. So, this is for region 7, that was for region 5. In region 7, there is also differences. But if you move to equilibrium state, you see more similarities. Okay. So this will give you a feel that what is the necessity of this more elaborate, detailed cell phenotype simulations as compared to the more simple and computationally less, less heavy phenomenological simulation. Okay? Unless you see these results and get a feel, then you get a feel of the whole thing. Just looking at equations will not trigger interest, isn't it? Equations are sometimes quite boring, isn't it? So I would stop. I hope you do not have any question at this point. Hello? Hitesh? Are we now?
Yes, sir.
I don't see the names of everybody. So whatever I see in front of me in a truncated form, I just call the names because I hope others don't mind. I calling a few names, I mean sometimes I try to wake you up. Okay? I hope everybody is awake.
Yes, sir.
Yes, sir. Yes.
I hope
Yes.
it was interesting to keep you awake.
Yes, sir. Yes, sir.
Yes, sir.
Or can I serve you some cup of tea?
That would be great, sir.
For your information and satisfaction, I have a news to give all of you. Okay. Yesterday, I had a long meeting with ISRO. A long meeting with ISRO, and today I have another one at 2 o'clock. By any chance, do you know Prithviraj Paul, one batch senior to you? Dual degree student? Last year, passed out last year. So it was a very important meeting, and this guy, Prithviraj Paul, is in a newly formed team in the Gaganayan project, where the cosmonauts, Indian cosmonauts, are scheduled to explore in the space, and they have to track biomechanics research on cosmonauts at ISRO. And they have identified me as the only person in the country who can help in developing biomechanics research at ISRO. They need to do kinematic analysis, as I have taught you, all of you, to do it. based on inverse dynamics, the motion capture system. Maybe they need it for the whole body. They may need it for the upper limb because they have to, at zero gravity or maybe different variation of gravity, the cosmonaut needs to operate machines. Machines means computer. Of course, machines are not the crude machines, but there could be a host of equipment that need to be operated at different gravity levels. And they need to simulate or need mathematical models of this kinematic model of musculoskeletal models of human being that needs to be simulated at different gravity levels. Plus, they need bone mechanics, stresses, strain, mechanical environment, and finally, remodeling. Okay? So when they were showing me their requirement, I was smiling and thinking of the students who are getting exposure to all the state-of-the-art topics. And there is none other who can give you in the country, none other to give you this level of exposure in biomechanics in one semester. So immediately you are thrown into the biomechanics world and you are ready to take up the challenge. The example is Prithviraj Paul. I checked yesterday, and before that, of course, 2 days ago, the email came it was a very urgent email. The director, the deputy director of the whole center in ISRO, they were urgently looking for developing this facility. And it's well, myself, I have done research about 25 years, but never thought about variation in gravity. Okay, it's all 1.0 gravity. Now, what will happen at zero gravity? I don't know. We have to investigate. Okay, at different levels of gravity, how will our motion system, the musculoskeletal system, how they will behave towards goal-directed movements is difficult to predict unless we actually start working on it using a systematic approach. So things are not easy. Let us see where it goes, but certainly I'm getting involved in the Gaganyaan project of the Indian cosmonauts or astronauts, whatever you say. Cosmonauts is probably a term used by a specific country, isn't it?
By Russia.
Russia, yes. So the Indian astronauts are planning to go to space, and this is a high priority project that needs biomechanical analysis of all sorts, whatever has been discussed in the class, starting from movement science, kinematic analysis, bone mechanics, bone adaptation, bone growth, everything. Bone regeneration, bone degradation. Okay. So continuous meeting. So we have a ISRO meeting at 2 o'clock scheduled. I just gave a 1 hour gap between my class and the meeting. But I need to also take a bit of rest and have lunch. By the way, Prithviraj Paul is our student, passed out in 2020. And yesterday he was in the meeting. I referred to the assignment that I've given all of you this year as well. And I said that you should actually give a presentation on HIP 98. That would be good for your colleagues in the office, that how this kinematic analysis is done and what needs to be actually implemented in the center, starting from a motion capture system that will give you the data of the kinematic data, and then using the inverse dynamics and further. So if somebody can understand HIP 98 well, the procedure, he or she will be quite conversant with the motion analysis system. Okay.